In this section, we are going to study early embryology, starting with gametogenesis. First, spermatogenesis, which is formation of sperms in the male. This occurs in the testis, which is made of seminiferous tubules. Here is a cut one. Now, in the wall of the seminiferous tubules, mitotic division of spermatogonia gives daughter spermatogonia. One enlarges to form a primary spermatocyte that divides into two secondary spermatocytes, which are haploid. These give two spermatids, which are haploid, and these transform into sperms. The process of transformation into sperms is called spermiogenesis. It happens that the cytoplasm becomes reduced, the nucleus is pushed to one side. From the other side, filaments will elongate to form the tail of the sperm. Here is a mature sperm. The head is made of a nucleus and an acrosomal cap. The neck or middle piece is made of axial filaments surrounded by mitochondria. The axial filament extends throughout the tail and is responsible for mobility of the sperm. Oogenesis in the female occurs prenatally. The cells in the ovary till puberty contain primary oocytes in arrested prophase. They are present in the ovary, as we see here, surrounded by granulosa cells and zona pellucida. Here they are. The follicles grow in size. And they acquire a fluid around them in a cavity called the antrum. The fluid is called liquor folliculi. The nucleus divides into two cells unequally, secondary oocyte and a first polar body. Ovulation occurs. The Graafian follicle ruptures and the ovum is released and picked up by the fallopian tube, waiting for a sperm to be fertilized. When fertilization occurs, the cell divides into two unequal cells, the mature ovum and the second polar body. Now we will study the process of fertilization in detail. Here are the sperms deposited in the female genital tract. They are deposited by hundreds of millions. They have to migrate up the whole very long journey up the female genital tract. This is a double sperm. This is a giant sperm. A certain percentage of abnormal forms like these are present in normal semen. The sperms move by the action of the tail. The tail looks like a whip or a lash that moves a rotatory movement that propels the sperm forwards. Many sperms die during this journey. Here they face the cervical mucus, which they penetrate to enter into the uterus from the vagina. Then they pass up the uterus. Many of them die during this journey. Then at the angle of the uterus they enter into the fallopian tube. Now we can see in the wall of the fallopian tube these microvilli and cilia. In the fallopian tube, a process called capacitation occurs to the sperms, in which they lose the glycoprotein coat which covers their heads and acrosomes. This process is essential so that sperms become capable of fertilization. They complete their journey till the lateral part of the fallopian tube, searching for an egg. Here they found the egg, waiting for about 24 hours to be fertilized. They try to penetrate the surroundings of the egg, which are the corona radiata, the zona pellucida, and the egg cell membrane. Many sperms manage to penetrate the corona radiata. However, when they come to the zona pellucida, only one sperm joins the membrane of the zona by receptor binding. This will make the acrosome membrane of the sperm head disappear with release of digestive enzymes that will help the sperm penetrate the zona pellucida. After this, the zona becomes impermeable to other sperms. Only one sperm manages to penetrate the zona pellucida. Now it binds the cell membrane, the vitelline membrane, penetrate it, and the vitelline membrane becomes also impermeable to other sperms. Here is the sperm losing its tail. Only the head enters. The zona reaction happens, preventing penetration of other sperms. The head of sperms makes the male pronucleus. The nucleus of the female makes female pronucleus. Both pronuclei fuse together, making the zygote or segmentation nucleus. Now, a process called cleavage starts, by which the cell immediately divides into two daughter cells or blastomeres, still surrounded by the zona pellucida. These migrate along the wall of the fallopian tube. Notice the cilia. The cells continue to divide within the zona pellucida into 4 cells, 8 cells, a group of cells called morula, which will then acquire a cavity, forming what's called blastocyst. All of this is still surrounded by the zona pellucida, which prevents increase of size of the dividing embryo. If the two cells separate, they can make identical twins. Now, the blastocyst enters the uterine cavity. Here it is, very small in size. Next, the zona pellucida should disappear by the action of enzymes of many sperms, and the blastocyst will be released from it. This process is called hatching of the blastocyst. Blastocyst will adhere to the wall of the uterus, the endometrium, which is now prepared by enzymes and is called decidua. Then the blastocyst will manage to penetrate through the wall of the endometrium to be embedded inside it, a process called implantation, as we will see later. Now we will study the process of implantation in a little bit of detail. Here, the trophoblastic cells or the outer cells make a syncytium which will penetrate. Now there will be differentiation first of the trophoblastic cells. The trophoblast will make a new layer called extraembryonic mesoderm. The extraembryonic mesoderm will acquire slits and a cavity called the extraembryonic coelom. Now we will study the development of embryoblasts. On the upper surface or dorsal surface of the embryonic disc, we will have the epiblast, which will acquire prochoral plate, cloacal membrane, primitive streak, and ending in a primitive knot. With elongation of embryonic disc, as we can see here, a depression in the epiblast or ectoderm is called neural plate. It will become deepened, forming a neural groove, which will give the development of the central nervous system, the brain, and the spinal cord. Now we will see the development of other layers. From the epiblast, from the primitive streak, this is the hypoblast below the epiblast. From the primitive streak, cells will grow ventrally, forming the endoderm, which will replace the hypoblast. Now again, more cells will grow from the primitive streak. Between the epiblast and endoderm, these will become the mesoderm. The mesoderm spreads between the ectoderm dorsally and the endoderm ventrally. Here is the ectoderm, and as we said, it will make the neural plate, which will give the brain and spinal cord. From the primitive node, a solid cord grows between the ectoderm and endoderm. This is the notochord, or the primitive vertebral column. It grows cranially all the way till the oral membrane. Now, finally, in the third week, a process called folding takes place, in which the embryo is folded upon itself. The amniotic sac becomes enlarged, the yolk sac becomes reduced and is connected to the intestine of the baby or the gut with the vitello-intestinal duct.